Good day, this is Professor Will, CBMD PhD. Today is Sunday, June 14th, 2020. It is 6:28 and 13 seconds a.m. Eastern Standard Time. And this is a continuation of my scientific research on bioinformatics. So let's get started here. Dynamic programming for global alignment. The classical global pairwise alignment algorithm using dynamic programming is the Needleman-Wunsch algorithm. In this algorithm, an optimal alignment is obtained over the entire lengths of the two sequences. It must extend from the beginning to the end of both sequences to achieve the highest total score. In other words, the alignment path has to go from the bottom right corner of the matrix to the top left corner. The drawback of focusing on getting a maximum score for the full line sequence alignment is the risk of missing the best local similarity. This strategy is only suitable for aligning two closely related sequences that are the same length. For divergent sequences or sequences with different domain structures, the approach does not produce optimal alignment. Dynamic programming for local alignment. In regular sequence alignment, the divergent level between the two sequences to be aligned is not easily known. The sequence lengths of the two sequences may also be unequal. In such cases, identification of regional sequence similarity may be of greater significance than finding a match that includes all residues. The first application of dynamic programming in local alignment is the Smith-Waterman algorithm. In this algorithm, positive scores are assigned for matching residues and zeros for mismatching. No negative scores are used. A similar tracing back procedure is used in dynamic programming. However, the alignment path may begin and end internally along the main diagonal. It starts with the highest scoring position and proceeds diagonally up to the left until reaching a cell with a zero. Gaps are inserted if necessary. In this case, a fine gap penalty is often used. Occasionally, several optimally aligned segments with the best scores are obtained. As in the global alignment, the final result is influenced by choice of scoring systems. The goal of local alignment is to get the highest alignment score locally, which may be at the expense of the highest possible overall score for a full-length alignment. This approach may be suitable for aligning divergent sequences, or sequences with multiple domains that may be of different origin. Scoring matrices. In the dynamic programming algorithm presented, the alignment procedure has to make use of a scoring system, which is a set of values for quantifying the likelihood of one residue being substituted by another in an alignment. The scoring system is called a substitution matrix. It is derived from a statistical analysis of residue substitution, data from sets of reliable alignments of highly related sequences. Scoring matrices for nucleotide sequences are relatively simple. A positive value or high score is given for a match, and a negative value or low score for a mismatch. This assignment is based on the assumption that the frequencies of mutation are equal for all bases. However, this assumption may not be realistic. Observations show that transition substitutions between purines and purines or between pyrimidines and pyrimidines occur more frequently than transversions. Substitutions between purines and pyrimidines. Therefore, a more sophisticated statistical model with different probability values to reflect the two types of mutations is needed. Scoring matrices for amino acids are more complicated because scoring has to reflect the physiochemical properties of amino acid residues, as well as the likelihood of certain residues being substituted among true homologous sequences. Certain amino acids with similar physiochemical properties could be more easily substituted than those without similar characteristics. Substitutions among similar residues are likely to preserve the essential functional and structural features. However, substitutions between residues of different physiochemical properties are more likely to cause disruptions to the structure and function. This type of disruptive substitution is less likely to be selected in evolution because it renders non-functional proteins. For example, phenylalanine, tyrosine, and tryptophan all share aromatic ring structures. Because of their chemical similarities, they are easily substituted for each other without perturbing the regular function and structure of the protein. Similarly, arginine, lysine, and histidine are all large basic residues, and there is a high probability of them being substituted for each other. Spiric acid, glutamic acid, and asparagine and glutamine belong to the acid and acid amylase groups and can be associated with relatively high frequencies of substitution. The hydrophobic residue group includes methionine, isoleucine, leucine, and valine. Small and polar residues include serine, threonine, and cysteine. Residues within these groups have high likelihoods of being substituted for each other. However, cysteine contains a sulfhydryl group that plays a role in metal binding, active site, and disulfide bond formation. Substitution of cysteine with other residues therefore often abolishes the enzymatic activity or destabilize the protein structure. The small and nonpolar residues, such as glycine and proline, are also unique in that their presence often disrupts regular protein secondary structures. Thus, substitution with these residues do not frequently occur. Amino acid scoring matrices. Amino acid substitution matrices, which are 20 × 20 matrices, have been devised to reflect the likelihood of residue substitution. There are essentially two types of amino acid substitution matrices. One type is based on interchangeability of genetic code or amino acid properties, and the other is derived from empirical studies of amino acid substitutions. Although the two different approaches coincide to a certain extent, the first approach, which is based on the genetic code or the physiochemical features of amino acids, has been shown to be less accurate than the second approach, which is based on surveys of actual amino acid substitution among related proteins. Thus, the empirical approach has gained the most popularity in sequence alignment applications. Empirical matrices, which include PAM and BLOSSOM matrices, are derived from actual alignments of highly similar sequences. By analyzing the probabilities of amino acid substitutions in these alignments, a scoring system can be developed by giving a high score for a more likely substitution and a low score for a rare substitution. For a given substitution matrix, a positive score means that the frequency of amino acid substitutions found in the dataset of homologous sequences is greater than would have occurred by random chance. They represent substitutions of very similar residues or identical residues. A zero score means that the frequency of amino acid substitutions found in the homologous sequence dataset is equal to that expected by chance. In this case, the relationship between the amino acids is weakly similar at best in terms of physiochemical property. A negative score means that the frequency of amino acid substitutions found in the homologous sequence dataset is less than would have occurred by random chance. This normally occurs with substitutions between dissimilar residues. The substitution matrices apply logarithmic conversions to describe probability of amino acid substitutions. The converted values are the so-called log odds scores or log odds ratios, which are logarithmic ratios of the observed mutation frequency divided by the probability of substitution expected by random chance. The conversion can be either the base of 10 or the base of 2. For example, in the alignment that involves 10 sequences, each having only one alignment position, 9 of the sequences are F, phenylalanine, and the remaining one I, isoleucine. The observed frequency of I being substituted by F is 1 in 10, 0.1, whereas the probability of I being substituted by F by random chance is 1 in 20, 0.05. Thus, the ratio of the two probabilities is 2, 0.1 divided by 0.05. After taking this ratio to the logarithm to the base of 2, this makes the log odds equal to 1. This value can then be interpreted as the likelihood of substitution between the two residues between 2 to the power of 1, which is 2 times more frequently than by random chance. PAM matrices. The PAM matrices, also called de Hoff PAM matrices, were first constructed by Margaret de Hoff, who compiled alignments of 71 groups of very closely related protein sequences. PAM stands for Point Accepted Mutation, although Accepted Point Mutation, or APM, may be a more appropriate term. PAM is easier to pronounce. Because of the use of very closely related homologs, the observed mutations were not expected to significantly change the common function of the proteins. Thus, the observed amino acid mutations are considered to be accepted by natural selection. These protein sequences were clustered based on phylogenetic reconstruction using maximum parsimony. The PAM matrices were subsequently derived based on the evolutionary divergence between sequences of the same cluster. One PAM unit is defined as 1% of the amino acid positions that have been changed. To construct a PAM1 substitution table, a group of closely related sequences with mutation frequencies corresponding to one PAM unit is chosen. Based on the collected mutational data from this group of sequences, a substitution matrix can be derived. Construction of the PAM1 matrix involves alignment of full-line sequences and subsequent construction of phylogenetic trees using the parsimony principle. This allows computation of ancestral sequences for each internal node of the trees. Ancestral sequence information is used to count the number of substitutions along each branch of a tree. The PAM score for a particular residue pair is derived from a multi-step procedure involving calculations of relative mutability, which is the number of mutational changes from a common ancestor for a particular amino acid residue divided by the total number of such residues occurring in an alignment. Normalization of the expected residue substitution frequencies by random chance and logarithmic transformation to the base 10 of the normalized mutability value divided by the frequency of a particular residue. The resulting value is rounded to the nearest integer and entered into the substitution matrix, which reflects the likelihood of amino acid substitution. This completes the log odds score computation after compiling all substitution probabilities of possible amino acid mutations. A 2020 PAM matrix is established. Positive scores in the matrix denote substitutions occurring more frequently than expected among evolutionary conserved replacements. Negative scores correspond to substitutions that occur less frequently than expected. Good day, this is Professor Will, CBMD PhD on infectious disease, bioinformatics, and microbiology. Well, hope you enjoyed listening. Until next time, have a great day.